So that's ion exchange chromatography. We'll talk about some examples of that in class. The resins for chromatography don't have to have pluses or minuses on them. There's other types of chromatography besides ion exchange. That resin could just have polar molecules, hydroxyapatite or silica. You used silica when you did TLC experiment in organic. And these would bind to polar biomolecules, and nonpolar things would flow through. Another type of chromatography we see less often in biochemistry is reverse phase chromatography, and it's the opposite of polar. Your resin is a really fatty alkane, in this case 18 carbons in a row, and it binds to hydrophobic molecules, and polar molecules flow through. This is seen some in lipid biochemistry in order to bind fatty lipids. Another type of resin that you can use is a gel filtration resin. This type of separation is also called size exclusion, or a type of dialysis. Now, in a gel filtration experiment, your resin, here shown here, does not have really any chemistry associated with it. It is just a bead, and that bead has holes in it of a particular size. Now, big proteins will be excluded from going inside the bead because they're too big to go through those holes, while smaller proteins will be able to go into the bead. In this example here, we've got a bead that has holes in it, a gel or size exclusion resin. The blue proteins here are too big to go in through those holes, while the red ones are able to jump in there. So if you put this resin onto a column and put a mixture, and the mixture in this case is big and small things, sort of counterintuitively, the big things will flow through more quickly. Now the reason why we get the big proteins off first is because their size excludes them from going into the beads. And so in order for the blue big protein to go from the start to the finish, they just have to travel through all the space in the column except for the space or volume occupied by the beads. That's a pretty small volume. The red protein, which can go into the beads, has to go through the entire volume of the column, including the beads. And so it takes the red protein much longer. And so the big things come out first, small things come out later. There's really no way to speed this up since there's no real chemistry associated with it. So you just keep dripping buffer through there until your small things elute off. Okay. This is also called dialysis, right? Not only are there resins with holes of particular sizes, but there are membranes. They look like bags, Ziploc plastic bags, that have holes in them of a particular size. And so if you have a mixture of big and small things, you can contain them in a dialysis tubing, put them in a big beaker with some buffer in it, and all the small molecules, small proteins will escape from your dialysis membrane because they can fit through the small holes, while the big proteins are retained within. You've heard of dialysis probably clinically. There's dialysis clinics all over, and they're used to remove small toxins from the blood. And so someone's blood is really pumped across these membranes to separate the small toxins from the bigger molecules that are put back into the patient.